Uh, Morning, everybody. Let's get started. So today we're going to start talking about the cholesterol metabolism and lipoproteins. All right. So first of all, cholesterol metabolism synthesis. So cholesterol is present in the body in actually two forms, right? The free cholesterol and cholesterol esters. Cholesterol esters, you guys know, right? You see one fatty acids molecules attached to the free cholesterol, forming cholesterol esters. So cholesterol, we need a cholesterol, right? So cholesterol has actually bad and good side. And the good side of cholesterol is that we need cholesterol because cholesterol is essential for the structure components of the cell membranes and or other layers of a plasma lipoproteins, right? Lipoprotein also has the monolayer membrane structures we're going to talk about later. And also cholesterol is the precursors of all other steroid and hormones like corticosteroids, steroids, and sex hormones, and bile acid, the vitamin D, and all these compounds actually are made from cholesterol, right? So cholesterol, and this is a good side of cholesterol. And we need cholesterol for cell membrane structures and for monolayer structures of lipoproteins, and also for synthesis of some important hormones. And the bad side, if we have too much cholesterol, we cannot metabolize it or get rid of it. And the bad side of cholesterol is that cholesterol is a major constituent of gallstones, right? You guys know, and also it's is the basis of the formation of atherosclerosis, right? So if we have too much cholesterol accumulate in like macrophages in the artery wall, then we're causing atherosclerosis and coronary disease and cardiovascular disease, right? So this is just sort of basic stuff of about cholesterol. So where actually our body's cholesterol comes from two major sources, right? Exogenous, major mainly from the dietary, from the diet, and also from endogenous that our body also can synthesize cholesterol. So about 50-50. 50% of our body cholesterol that you have from diet, and another 50% from synthesis in the body, right? De novo synthesis of cholesterol. And for the biosynthesis of cholesterol, it's 10% of the total cholesterol in the liver. Sorry, what I'm trying to say is that 10% of the total synthesis of cholesterol actually occur in the liver and intestine, right? So in other words, that the liver and intestine are the major, or the most important site for cholesterol synthesis, right? And in terms of cells, almost all type of cells are able to synthesize cholesterol, right? And the location inside of cells where actually cholesterol synthesized is actually synthesized in the ER or cytosol, and it's synthesized from acetyl-CoA. So acetyl-CoA again is a precursor of a cholesterol synthesis. And transporter, in terms of transporter, cholesterol is a lipid, right? So it's a transporter requires the lipoproteins, and two types of lipoprotein are the major transporters of the cholesterol, LDL and HDL. So LDL basically is help uptake of or to deliver cholesterol into the peripheral cells, into the cells, right? Deposit cholesterol in the cells. And HDL, the basic is a good cholesterol, the transporter cholesterol out of cells and delivered to the liver for excretion, right? So LDL, usually people call it bad cholesterol, and HDL, good cholesterol, right? Because of the functions different. LDL, you remove cholesterol from the cells and from the body, and LDL is deposit cholesterol into the cells, right? And HDL actually removes the extra cholesterol from the peripheral cells and get rid of to the liver and get rid of the extra cholesterol. Okay, now let's talk about the synthesis of cholesterol. So synthesis of cholesterol also is the complex process, involve many steps, reactions, but you don't need to remember each reactions. Again, usually try to remember the key steps and key enzymes that control this synthesis pathway. And the substrate or precursors of cholesterol, right? You need to know. Again, the precursors of cholesterol synthesis is acetyl-CoA. Remember which other compound that also synthesized from acetyl-CoA? Ketone bodies, right? We just talked about last class. So cholesterol also requires acetyl-CoA for synthesis. And this two steps, the initial two steps in cholesterol synthesis are common to both ketone body synthesis and cholesterol synthesis, right? So the first initial step is the same as the ketogenic pathway, right? So start with the acetyl-CoA molecules, the two acetyl-CoA molecules being condensed to the acetyl-CoA, right, by silase, and the third acetyl-CoA being added to the acetyl-CoA molecules and forming HMG-CoA, right? So the two steps actually the same step in both ketone body synthesis and the cholesterol synthesis, right? And requires acetyl-CoA molecules. And in general, so I'm going to divide the multiple steps in the cholesterol synthesis into like a major 5 steps, right? So the first, major step is called synthesis by veronate, right, from 3 molecules of acetyl-CoA. So 3 molecules of acetyl-CoA is through the several reactions. We're not going to talk about it, to form the veronate, right? And in this series of reactions, the enzyme HMG-CoA reductase is the key enzyme, right? It's also the whole process, the key enzyme for the whole process of cholesterol synthesis. So try to remember HMG-CoA reductase is the key enzymes, right? And it also, okay, so this is the key steps, right? See how HMG-CoA reductase catalyzes the reaction, the formation of mavoronate. So HMG-CoA being reduced to mavoronate, right? And requires a 2 NAD pH. And you see, and also you try to remember, what kind of factors that can regulate HMG-CoA reductase and then control cholesterol synthesis pathways, right? That involve the bile acid, cholesterol levels itself, and mavoronate levels, right? These 3 things all can inhibit HMG-CoA reductase activities and slow down or inhibit the cholesterol synthesis pathways, right? Which makes sense, right? If we have too much cholesterol already in the body, then we don't need synthesize, right? So you need to inhibit HMG-CoA reductase to reduce the cholesterol synthesis, and mavoronate is also the intermediates the cholesterol synthesis pathways, right? So increased mavoronate levels also indicate that you have high level of synthesis activities, you also, will try to slow down the pathway so it inhibits HMG reductase, and bile acid also inhibits the mavolinate synthesis. And the other factors is actually JAK is stacking, is the cholesterol lowering JAK, and the mechanism for controlling cholesterol levels is also by inhibiting the HMG-CoA reductase activity, right? So this enzyme is really interesting, important JAK target, right? So you design the JAK to inhibit this enzyme activity and to reduce the de novo synthesis of cholesterol and eventually reduce the cholesterol level, right? Okay. So in step 2, the formation of a isoprenyl diphosphate. So mevalonate be converted to an isoprenyl diphosphate through the three sequential phosphorylation reactions and one decarboxylation reaction, right? 4 reactions involved in this conversion, and 3 ATP is required. And here are the 4 enzymes that are involved in this step, called 3 kinase, mevalonate kinase, phosphomevalonate kinase, and the diphosphomevalonate kinase, right? Basically, catalyze the phosphorylation. So kinase I mentioned before usually is enzyme that catalyze the phosphorylation reaction, right? And the diphosphomevalonate decarboxylase, the 4 enzymes. You just don't need to memorize these enzymes. Step 3 is the secreting formation from isoprenyl diphosphate. Is also multiple steps involved, right? Isomerization, condensation, reduction reaction, right? And requires NADPH for the reduction reaction, right? Okay, step 4 is the conversion of secretin to melanosterol, right? So the 2 enzymes is involved secreting apple oxidase and oxidase secreting melanosterol cyclase, right? Just making the cyclic structure of the cholesterol. Okay, so the last step is the generation of cholesterol from how to pronounce it? I don't know. Do you guys know how to pronounce this? Try to be lanosterol. Lanosterol. Okay, I'm trying to make sure I pronounce it correctly. Okay. So, and this is isomerase, and delta 24 reductase that are involved, right? Again, it also requires NADPH for, for proton for reduction, right? So, generate double bonds in the cholesterol structure. So, cholesterol structure has one double bond here, right? Okay, so this is the whole process. The 17 enzymes that involved. And again, the substrate, a precursor, the acetyl-CoA, and requires 8 NADPH and 3 ATPs, right? So, and synthesize this very complex, structures of cholesterol. So the key enzyme, right? And try to remember HMG-CoA reductase, and the factors that regulate this enzyme activities. We talk about the 3 and 4, right? Can you guys remember? which 4 factors that can regulate these enzymes? Yes. Statins, yeah, it's a drug, right?
Like cholesterol,
Right. Uh-huh. Bile acid, right? Bile acid and veronate and cholesterol, right? So 4 negative regulators of HMG-CoA reductase. Okay, now let's talk about the regulation of cholesterol homeostasis. Homeostasis means that the balance you need to be maintained between the synthesis of cholesterol and the disposal, right? We wanted to keep balance, we wanted to maintain the just right amount of cholesterol in the body. We don't want to have too much cholesterol, right, in the body. We know the bad side of cholesterol. So the regulation basically regulates the synthesis or the deposit, right? The two sides, then we need to regulate them so then can control the normal levels of the cholesterol. So the regulation of cholesterol synthesis and regulation of cholesterol uptake that are involved in LDL receptors, and also the called reverse transport of cholesterol by HDL is also one of the important regulate mechanisms, right? So sorry I didn't include in here. So actually 3 aspects in terms of the regulation. First, at the synthesis level, right, we regulate HMG-CoA reductase enzyme to regulate synthesis of cholesterol. And the second, regulation of cholesterol uptake by LDL. We talk about LDL, right? LDL basically transport cholesterol into the cell. So this point is also very important, need to be regulated. And the third regulation is the cholesterol removed, by HDL, right, from peripheral cells back to the liver and excreted by bile acid, biosynthesis, through the synthesis of bile acid, right? So 3 levels of regulation. So any steps in this 3 regulation that can causing this or imbalance of the cholesterol in the body and causing some health problem. So this is just a general regulations, and here basically cholesterol synthesis, right? And this is a key enzyme, and you see the hormones and the substrates can all regulate this enzyme activity. And we already talked about the mevalonate and cholesterol, then as statins like inhibits the HMG-CoA reductase, and also bile acid, right, inhibits HMG-CoA reductase. And also 3 more things like starvation. During starvation, this enzyme activity is also reduced, so cholesterol synthesis also decreased. And glucagon and glucocorticoids and also inhibitors of the HMG-CoA reductase. And insulin and thyroid hormone are the positive regulators, right? These 2 hormones can stimulate HMG-CoA reductase and increases the cholesterol synthesis, right? So this is the 1 level of regulation of the cholesterol homeostasis. And here I just wanted to talk about a little bit more detail how cholesterol regulates the HMG-CoA reductase, right? And a little bit involved like molecular mechanisms, right? So both cholesterol and mevalonate can inhibit this transcription factor called sterol regulatory and element binding protein, right? SIBP. So SIBP is the transcription factor that controls the transcription of HMG-CoA reductase enzyme, right? So what cholesterol and the veronate does is to inhibit binding of SREBP protein to the DNA sequence, right? So SRE called stereoregulative element, basically the DNA sequence that located upstream of HMG-CoA reductase enzyme coding sequence, right? So when cholesterol or veronate levels increase, then inhibits binding of SREBP to SRE DNA sequence, right? And then inhibits the transcription of HMG-CoA reductase, right? So you just need to remember that this transcription factors involved in the mechanism, right? So these two substrates directly interact with the or interfere with the SIBP binding, right? SIBP function in transcription of the HMG-CoA reductase. Okay, so and in terms of the other two aspects, regulation, like LDL uptake cholesterol or HDL, the reverse transport, we're going to talk about later, and when we talk about lipoprotein lipase, right? So just keep in mind that three, levels of regulation is important to maintain the cholesterol stasis. Okay, now we switch to the topic lipoproteins, right? So lipoprotein is basically a complex of lipids and proteins, right? We mentioned before, and the lipids, cannot be freely present in the plasma. It cannot be freely transported from different, between tissues. So that requires a protein to help the transport lipids, right, between tissues. So lipoprotein is the most important systems or complex, the lipids and protein that help the transport of lipids, right? So the main function of lipoprotein is to transport lipids from tissue to the tissue in the plasma, in the circulation, right? So again, the lipoprotein basically present in the circulation, right? So there are actually 5 different type of lipoproteins that are present in the circulation. Chymicron, VLDL, IDL, LDL, HDL, right? And different lipoproteins have different size and different composition of protein and lipids, right? And so chymicron is actually the largest lipoprotein and lowest in density because of the ratio, the liver lipids versus the lipids to the protein, right? Is high. So that means the chymicron has the most abundant lipids, right, compared to other lipoproteins. So that makes the chymicron lighter, right, compared to other lipoproteins. And it also contains the highest percentage in terms of weight of triglyceride, right? So chymicron, and actually chymicron and VLDL are considered as a triglyceride lipoprotein because they have a very high percentage of triglyceride. Or in other words, these two types of lipoprotein, the main function is to transport a triglyceride, right? And VLDL is called a very low density lipoprotein. It's the second largest, highest in the triglyceride the content. So again, I already mentioned, right, this chymicron and VLDL mainly the main function is to transport the triglyceride. It's a triglyceride-rich lipoprotein. And IDL, intermediate and density lipoprotein, is actually the remnant of the VLDL And LDL is a low density lipoprotein, and HDL, the high density lipoprotein. So LDL and HDL have most abundant cholesterol esters, right? Cholesterol content. So this LDL and HDL, the function of these two lipoproteins are to primarily transport cholesterol, right? And here I show that the LDL and HDL, the most abundant lipoproteins in the plasma, because these two lipoproteins, the size is relatively small, and the can stay longer, right? So the turnover rate becomes low, but the chylomicron is the largest, lipoprotein, and turn rate very faster. So because the turn rate is slow, so they stay longer in the circulation. So that's why these two small lipoproteins are more abundant, right, in the plasma. Okay, now about the structure of lipoproteins. So lipoprotein basically like a ball, and inside has the core lipids, and the surface, the lipid surface. Lipids basically are the monolayer of phospholipids, right? So the lipid core it contains contains the most important lipids that we transport, right? So basically, as the cholesterol esters and the triglyceride, right? So a lipid core contains the both triglyceride and the cholesterol esters, and on the surface lipids and monolayer of phospholipids, and also free cholesterol, right? Free cholesterol on the surface membrane and make the structure of the surface membrane, right? And in addition to the lipids, there are also the protein component, right? And basically localized on the surface of the lipoproteins. So all these proteins we give them name called apoproteins. Right? So different type of apoproteins that localize on the surface of the lipoproteins, and the apoprotein also have own important functions we're going to talk about later. And different lipoproteins at different type of apoproteins on the surface, right? Let's see. All right, so so let's talk about apoproteins. We know that apoprotein basically is on the surface of the lipoproteins, and in general we have the 4 different types of apoprotein: apoprotein A, B, C, and E, right? And the functions of apoprotein. So first of all, the apoprotein that can be as a cofactors for enzymes, for enzyme activities. An example for this is ApoC2. Try to remember this apoprotein, right? ApoC2 is the important apoprotein that is for lipoprotein lipase activities, right? So the ApoC2 on the surface of lipoprotein will help lipoprotein to track to the lipoprotein lipase, so allow the lipoprotein lipase to catalyze the hydrolysis of a triglyceride, right? And maybe it will also help you to understand. You know that lipoprotein lipase mainly hydrolyze triglyceride, right? In which lipoprotein? Chylomicrons and the VLDL, right? So So then probably also help you to understand that ApoC2 basically mostly localized in the chylomicron and VLDL, right? Because it requires for the lipoprotein lipase to hydrolyze triglyceride. Okay, so the second function of the lipoprotein is the structure integrity. So lipoprotein is also is stabilize lipoproteins, right? The apoprotein stabilize lipoproteins. And third is some of the lipoproteins are the important ligands for receptor bindings, right? Also try to remember this ApoE, right? So ApoE is the protein, the lipoprotein that help the lipoprotein to recognize the receptor LDL receptor and LRP is the LDL receptor related protein, right? So LRP is LDL related protein. So ApoE on the surface of lipoproteins that help this protein to the whole complex to recognize the receptors and for internalize the whole lipoprotein into the cells, right? So LDL receptor is very important and localized on the cell surface, particularly liver, and recognize the ApoE contains protein, lipoproteins, and take whole lipoprotein into the cells, right? And the last important function of protein is to make the suspension of lipids in the plasma, right? So I think that it's two important things in ApoC2 and ApoE particularly function. Try to remember these two, particularly apoprotein, right? All right, composition of lipoproteins. Then we talk about specific lipids and protein composition in each lipoproteins. So you see, got core lipids mean that the lipids within the inside of a lipoprotein, and this core lipids basically is the lipids that need to be transported, right, from tissue to tissue, and surface lipids just make the structure of the lipoproteins. So core lipids we know that contains the cholesterol esters. CE means cholesterol esters, represent cholesterol esters, and TG triglyceride, right? And surface lipids, free cholesterol, and LPL is phospholipids, right? And what kind of lipoproteins on the each different protein. So let's look at the chylomicron first. So chylomicron, you can see both actually chylomicron and VLDL we already talked about, right? The triglyceride-rich chylomicron. So means that the core lipids contains the majority of the core lipids are triglyceride. So chylomicron, you see core lipids at about 80 to 90% of core lipids are triglyceride, and in VLDL, 50 to 65% of core lipids are triglyceride, right? And also contains some cholesterol esters. And also try to remember the apoproteins in these two different apoproteins. So ApoB40 and ApoB100 are unique to each chylomicron and VLDL, right? So ApoB48 is basically synthesized in which tissues and organs? Remember? Yeah, small intestine, right? And what about the chylomicron? Where's the chylomicron synthesized? Also a small intestine, right? So you connect like ApoB48 to chylomicron, right? So both chylomicron and ApoB48 are synthesized in the small intestine. So ApoB48 is unique to chylomicron, right? And ApoB100 is basically synthesized in the liver, right? And VLDL is also mainly synthesized in the liver. So, so ApoB100 is unique to VLDL. And then look at the LDL and HDL. And these are the two lipoproteins that cholesterol-rich lipoproteins, right? You can see the 35 to 40% of core lipids, the cholesterol esters, and for LDL, and 14 to 18% of core lipids and cholesterol esters for HDL. And LDL, pretty simple, only contains the surface at the ApoB100. Easier to remember. HDL and ApoA1 is also unique to HDL. Right? And our IDL basically is a remnant of a VLDL, right? And it contains both ApoB100 and ApoE, which is related to VLDL, right? Since it's remnant of the VLDL. Right? And people also, well, I can talk this later. So. Question? Yes.
Yeah, on the previous slide with functions, did we say ApoE is on LDL?
No, the apoprotein always on the lipoprotein, the complex, right? Meaning that the LDL receptor that can recognize ApoE proteins, so can check the ApoE containing lipoproteins, and then take up like ApoE containing lipoprotein into the cells. We will get there, right, with the detail. Okay, so remnant lipoprotein. Remnant lipoprotein means that after a hydrolysis of the lipids in the lipid core, that the lipoprotein becomes smaller because you're losing the lipids, right? So the product is called remnant. So for example, the chylomicron, we transported triglyceride from the diet, right, which absorbed it, in the small intestine and to the adipose tissue. So then the I think I probably opened the wrong file. Anyway, so LPL in the adipose tissue, LPL is released, right, from the adipose tissue, and hydrolyzed triglyceride in the chylomicron, in the core, right, of chylomicron, and take up, and the triglyceride being break down fatty acids and taken up by tissue, and this is basically the adipose tissue, right, in the fat condition. And then chylomicron then become chylomicron remnant. And so if you compare to the chylomicron remnant, thinking about the lipid composition or lipid is how different is the lipid composition between chylomicron and chylomicron remnant. So chylomicron remnant should have much less triglyceride, right, compared to the chylomicron, because about the 90% of the triglyceride in the chylomicron being already taken up by adipose tissue, right? So the chylomicron remnant, which still contain some triglyceride and being picked up by the liver. And VLDL, similarly, the transport triglyceride, which is synthesized in the liver to the adipose tissue, and it's again by LPL, hydrolyzed triglyceride in VLDL, and fatty acid being picked up by adipose tissue or other tissue like muscle, and VLDL become remnant, and IDL and LDL, right? So IDL also have much less triglyceride compared to VLDL, right? Is that clear about lipoprotein and remnant lipoproteins, right? And but not only the lipid composition is changing, the surface apoprotein is also changed, right? We will talk about this later. So you're just thinking about what is difference between chylomicron remnant in terms of the lipid composition or the protein composition, right? Apoprotein. Okay, so now I'm going to talk about each chylomicron, right? Go through in terms of the composition and function. So first, lipoprotein is chylomicron. So tell me again the core lipids in the chylomicron. What type of lipids in the core of chylomicrons? Triglycerides, right? It's a major lipids in the core of the chylomicron. And what else? Cholesterol esters, right? So cholesterol esters and triglyceride and cholesterol esters, and again the 85 to 95% actually lipids are triglyceride, right? And apoprotein, what kind of apoprotein that present on the surface of the chylomicrons? ApoB48, right? This is specific. So try to remember ApoB48, and it's specific or unique to chylomicrons. And synthesis. So where are chylomicrons synthesized? Small intestine, yes, the enterocytes. And where does triglyceride in chylomicrons come from? Diet? Yes. Great. Thank you. So diet, right? And ApoB is made from small intestine enterocytes. So it's unique to chylomicrons, right? So what is the function of chylomicrons? What is the function? What does chylomicron do? Synthesize in the small intestine. Yes, so transport the dietary lipids to the tissues, right? Okay. Before we start to talk about this could have a long explanation, so let's take about 10 minutes break, right? now let's move on. So I think I will just talk about the chylomicron, right? Now let's get to the metabolism of chylomicron, how chylomicron metabolize, right? We know that the purpose of our body synthesize chylomicron is to transport lipids, right? Dietary lipids from small intestine to the other tissues. And this process is mostly occur in which physiological condition, or in other words, in which physiological conditions chylomicron is synthesized and need to be synthesized. In the fat condition, right? So, and the purpose of chylomicron synthesis is to transport dietary lipids from small intestine to which tissues you think will be the major tissue that, no. I mean, in the fat condition, right, the chylomicron is to transport dietary lipids. In the fat condition, what our body needs to do is to store energy, right? So adipose tissue should be the major site or the target tissue that chylomicron transport lipids to. So, so this sort of the whole process. Now we know that chylomicron synthesized in the small intestine, and once it's synthesized, it's called the initial chylomicron synthesized in the small intestine is called nascent chylomicron, means that the chylomicron actually only contains the B48, which is synthesized from the small intestine, right, and ApoA, but does not have the other type of apoproteins, for example, ApoC, ApoE, right? So, and ApoC, ApoE basically are synthesized in the liver, right, and transferred from HDL to the chylomicron, right? So once chylomicron receives the ApoE and ApoC from HDL, which initially synthesized from the liver, then become the mature chylomicron, right? So mature chylomicron has lipid core and every type of apoproteins, including B48, ApoE, ApoC, and A. Right? And we know that the final destination of chylomicron is adipose tissue. Right? So this peripheral tissue, you can put adipose tissue there. So, and we also talk about, let me see, I'm just kind of curious. Actually, I forgot to put the ApoC2 in here. So, so in the adipose tissue, we know that the main thing that is to store triglyceride, right, in the adipose tissue that initially from the diet. So triglyceride in the chylomicron, first of all, need to be break down, right, by LPL, which is synthesized and secreted from adipose tissue. And remember we mentioned earlier that one of the important apoproteins, which one is important for the lipoprotein lipase activity? You tell me? ApoC2, right? Remember. So ApoC2, which is on the surface of a chylomicron, is required for the LPL to target the chylomicron and to hydrolyze triglyceride in the chylomicron and release fatty acids, which can be uptake by adipose tissue or some other tissue as well, but mainly adipose tissue in the fat tissue, right? And also release the glycerol. Glycerol can go back to the liver for this at this stage was not for gluconeogenesis, right? And then chylomicron become chylomicron remnant. So you see the size of the lipid core is much smaller, right, compared to chylomicron. That means a lot of 90% of triglyceride already been break down and uptaken by adipose tissue. And the chylomicron remnant, and also missing the other two apoprotein, ApoA and ApoC, which actually recycle back to or transfer back to the HDL. Right? So ApoA and ApoC is called transferable apoprotein. They always transfer between the two different lipoproteins. So chylomicron remnant, which has a ApoB48 and ApoE, can be recognized by the receptors, LPL receptors, right, on the surface of the liver. So because of ApoE, right, so can be recognized ApoE on the chylomicron remnant by LDL receptors, and then take up by the liver, right? So the chylomicron remnant eventually is picked up by the liver and mediated by LDL receptors. And this process requires ApoE, right? Requires ApoE on the chylomicron remnant. So in some cases, if ApoE is deficient or ApoE kind of mutation, then the chylomicron remnant cannot be picked up by the liver, right? Because LDL receptor cannot recognize the chylomicron remnant if it does not have an ApoE on the surface. All right, so it seems like simple, right? For the metabolism, chylomicron again it occurs in the fat condition, and the purpose of chylomicron synthesis is to transport dietary lipids from small intestine to primarily adipose tissue for storage as a triglyceride, and also remnant can be picked up by the liver, right? So in the liver, if you still remember what kind of enzyme that hydrolyzes triglyceride in chylomicron remnant? Yeah, hepatic lipase, right? So liver has its own lipase called hepatic lipase that hydrolyzes triglyceride in chylomicron remnant, right? And break down the triglyceride to the fatty acids. And this fatty acid being reacidified back to triglyceride and packed into VLDL, right? VLDL is a major lipoprotein synthesized in the liver, right? And the VLDL being released again, and then picked up by other tissue, including adipose tissue. So, right? Any questions about the chylomicron metabolism? Is that clear? So, the last thing is the fatty acids being picked up from chylomicron primarily goes to the synthesis of triglyceride in the adipose tissue, right? And then stored in the adipose tissue. So this sort of a simplified version of the chylomicron metabolism. The mature chylomicron, you can lipoproteins, all types of lipoproteins, being first of all, transported to the adipose tissue, and triglyceride, 90% of triglyceride in chylomicron being hydrolyzed by LPL, which is released from adipose tissue, right, and to fatty acids, glycerol, and pick up fatty acids pick up by adipose tissue. And in the meantime, the mature chylomicron also lose the ApoA and ApoC, and eventually become chylomicron remnant. Chylomicron remnant being picked up by liver through the mechanism, right, LDL receptor mediated mechanism. Again, the ApoE is required for the LDL receptor binding and recognizing binding and pick up by the liver and hydrolyzed by hepatic lipase in the liver. So here are the two questions. So which enzymes hydrolyze triglyceride in the chylomicron? Depend on the tissue, right? In adipose tissue, it's LPL. And which enzymes hydrolyze triglyceride in chylomicron remnant? Hepatic lipase, right? Okay. Right. Now move on to VLDL. Core lipids in VLDL. What are the core lipids in the VLDL? Triglyceride and cholesterol esters. Again, VLDL also is the triglyceride rich, the lipoprotein, right? And apoproteins. What type of apoproteins on the surface of VLDL, or which apoprotein synthesized in the liver? B100 and ApoC2 and E are also on the surface of the VLDL, right? So ApoB100 is specific liver. And synthesis, where are VLDL synthesized? Which tissue? Liver is the place where VLDL synthesized, right? And where do triglyceride in VLDL come from? So that means what type of triglyceride, what sources of triglyceride VLDL actually transport? Three different sources of triglyceride can be packed into the VLDL and transported, right? First is the de novo lipogenesis, which you guys know, right, from dietary carbohydrate. So this basically occurs in the fat condition, right? We eat the dietary carbohydrate and be converted to fatty acids in the liver, right, through the lipogenesis pathway. And second source is long-chain fatty acids internalized by the liver from plasma protein, lipoprotein remnant. We just talked about, right? Chylomicron remnant can be picked up by the liver and break down to the fatty acids. So, and this fatty acids or triglyceride initially from the diet, right? And the third sources of triglyceride in VLDL is unacidified or free fatty acids. So free fatty acids, can you thinking about which tissues are the major sources of free fatty acids? Which tissues release adipose tissue? Yes, and in which physiological condition? that adipose tissue release fatty acids. Fasting, right? So the third sources of fatty acids actually mainly come from adipose tissue under the fasting condition. So fasting condition, so in other words, the VLDL can be synthesized in both fat and fasting condition in the liver. Right? In the fat condition, VLDL basically transported triglyceride from diet or from the conversion of carbohydrate to the fatty acids, right? And the destination for VLDL transport of these sources of fatty acids in the fat condition is to adipose tissue, right? For storage in the fat condition. But under the fasting condition, VLDL is synthesized, and the majority of the triglyceride that VLDL needs to be transported from adipose tissue, right? During the fasting, we have lots of fatty acids being released, and liver picks up and synthesizes triglyceride, pack into VLDL, and to which tissues? Yes, muscle during the fasting condition, right? Muscle needs to utilize fatty acids, right? So, and fatty acids be packed into VLDL and released from the liver and transported to muscle, right? Very good. And ApoB100 is synthesized in the liver. Right, so now let's go over this, the process of VLDL metabolism. So you all know, right, what is function of VLDL just described. It depends on the different state, fat or fasting, right? And okay, so let's start with the liver. So liver is the major site for the synthesis of VLDL. So VLDL is synthesized in the liver and packed to triglyceride and transported to the other tissues. And nascent VLDL also lacks the ApoC and ApoE. Although have some ApoE, ApoC can be synthesized, in the liver, but they need more ApoE and ApoC from transferred from HDL, right? So HDL can also give the more ApoC, ApoE to nascent VLDL and makes it become mature VLDL, right? So it has enough, high enough levels of ApoC, particularly ApoC2 on the VLDL, so it can be recognized by lipoprotein lipase, in which released from adipose tissue, right? And again, ApoC2 is required for the activity of LPL, so hydrolyze triglyceride, release fatty acids, and pick up by adipose tissue in the fat condition, right? So I've made actually 2 more slides to describe a little bit more detailed in terms of VLDL function in fat and fasting condition, right? And here we just specifically talk about, in the fat condition, right? Or if you wanted to write a fat in this corner, so just, and fat condition, the most of triglyceride, right, being picked up by adipose tissue, right? And after, the pick up triglyceride by adipose tissue, VLDL then become VLDL remnant, which is IDL. So IDL also have a smaller size and have much less triglyceride in the core, and also losing ApoC, right? So only have ApoB and ApoC, ApoE remain on the surface, the LDL. And IDL can be recognized by LDL receptor because of ApoE on its surface, right? So, and I think about 80% of IDL is picked up by the liver, and the 20% of IDL can become LDL, right? So LDL is also sort of an exchange, lipids exchange between the different lipoproteins occurred. We will talk about a little bit later, but here I just want to mention, see the composition of lipids actually change, right, from IDL to LDL, because there is lipid exchange happening between HDL and LDL, means that LDL will receive the cholesterol from HDL, but give the triglyceride to the HDL. So after this lipid exchange, the LDL become more cholesterol rich, right, and have a less triglyceride. So LDL also the ApoE is lost, right, from the LDL surface. So it only contains the ApoB 100, and this LDL also can be picked up by the liver, but also by the called extrahepatic tissues, right, including the artery wall, and macrophages on the artery wall, and which is a bad thing, right? If we pick up too much cholesterol from LDL, particularly in the macrophages, see this, the artery wall, and lots of macrophages pick up the LDL cholesterol, and LDL cholesterol accumulate in macrophage and form so-called atherosclerosis. What is the formation, right? Okay, so actually the most of the LDL being picked up by the liver because of the LDL receptor is most abundant in the liver, and also all the other tissues has LDL receptor. So this is the major pathway, right? LDL pick up by liver, and this sort of the not a major pathway, but it still have pick up by other tissues from the LDL. Okay. Any questions about this? Yes.
Going back a couple of steps, you're talking about LDL being recognized by adipose tissue and ApoC2. What was the ApoC2?
ApoC2?
Yeah.
ApoC2 is required for LPL function. Right? So ApoC2 sort of can recognize by this enzyme. So the enzyme be able to target this VLDL particles and to hydrolyze triglyceride inside of the particle. Right? Questions? Okay, simplified version of the VLDL metabolism. The VLDL synthesized and transported triglyceride to the adipose tissue in the fat condition, and in the meantime then loses ApoC and become VLDL remnant, which is IDL, and IDL being picked up 80% by the liver through the LDL receptor mechanism, and again ApoE is needed for the recognition of LDL receptor, right? And 20% actually become the LDL. So LDL eventually also, the ApoE is gone, right, and from the LDL. So LDL can either pick up by the liver and also peripheral tissue, right? If picked up by the macrophages in the artery wall and causing some health problem like atherosclerosis, and also the sort of exchange of lipids between HDL and IDL. Right? So, so make the LDL more cholesterol ester rich and lipoproteins. Okay. Now LPL, I just wanted to emphasize this enzyme again to talk about it a little bit more, give more information about this enzyme, right? First of all, synthesized mainly, you guys already know, by which tissues? Adipose and muscle, right? Adipose tissue and muscle are the major tissue that synthesize LPL, and this enzyme actually located on the wall of a capillary endothelium within the tissue, right? So when the blood supplies it goes into the tissue and carry this, like protein into the tissue, and this enzyme will start to have function, right? And it's not in the liver. So liver has its own lipase, right, which is called hepatic lipase. And the function of this enzyme is to hydrolyze triglycerides, release fatty acids, and that can be transported into the tissue. And different tissues, here we talk about adipose tissue and muscle, although they all can produce LPL, but they have a different activities, or in other words, the activity being regulated differently, right? Depend on the physiological state. So, for example, the LPL have a low Km for triglyceride in muscle and high Km for triglyceride in adipose tissue. And main thing is that during the fasting, the LPL for activity of the triglyceride hydrolysis is high in which tissues? Muscle, right? And in fat condition, LPL activity is high in the adipose tissue, right? And we also have a new information we learned from today that LPL activities require the presence of ApoC2 on the lipoprotein, surface of lipoprotein, right? Particularly chylomicron and the VLDL, right? And insulin regulates the synthesis and secretion of LPL from which tissues? What do you would expect? Adipose tissue, right? Okay. Okay, so the next two slides are new. I'm going to post it. Don't worry about it. I just wanted to clarify some information. It's probably kind of confusing in terms of the LPL activities, right, in different tissues, in different physiological state. So LPS in adipose tissue. We know that LPS in adipose tissue is increased in the fat condition by insulin stimulation, right? And so in fat condition, we know that the chylomicron is synthesized, right, and try to transport the dietary fatty acid triglyceride to the adipose tissue for the storage. And in chylomicron, so in the fat condition, in adipose tissue, LPL has two substrates, right? One is the chylomicron, triglyceride in chylomicron, and the other is triglyceride in VLDL. So LPL in the adipose tissue, LPL can hydrolyze triglyceride in both chylomicron and VLDL, right? And the triglyceride in chylomicron, and it's from the diet, right? So triglyceride in chylomicron basically dietary lipids, and triglyceride in VLDL it's also actually there's the dietary lipids, but it's a little bit different. I just wanted to clarify a little bit more. So in VLDL is synthesized in the liver in the fat condition, right? The fatty acids can come from dietary carbohydrate through the lipogenesis, and fatty acid can also be coming from chylomicron remnant. Remember, right? Chylomicron, majority of chylomicron being, go to the adipose tissue and pick up triglyceride, but still have, become chylomicron remnant. So chylomicron remnant eventually go to the liver, right? So this fatty acid could come from conversion of dietary carbohydrate or come from the chylomicron remnant, which initially from the dietary lipids, right? So just try to remember the LPL in the adipose tissue is activated in the fat condition, stimulated by insulin, and hydrolyzed triglyceride in both VLDL and chylomicron. And this triglyceride basically initially from the diet, right? Now in the muscle, so muscle or during the exercise, the muscle can also release or produce the LPL, and the major substrate for LPL in the skeletal muscle is VLDL, right? The VLDL. And the VLDL is secreted from liver, and the fatty acids for triglyceride synthesized in the VLDL primarily comes from adipose tissue, right? So during the fasting or exercise, catecholamine like epinephrine or norepinephrine, particularly epinephrine, increase, and which can stimulate lipolysis, right? Stimulate the hydrolysis of triglyceride and release free fatty acids and increase fatty acids level in circulation, and this fatty acid be picked up by the liver and converted back to the triglyceride and packed into VLDL and transported to the skeletal muscle, right? For the so LPL is also released synthesized from skeletal muscle and break down triglyceride and release fatty acids and pick up by the skeletal muscle. And try to remember, in both fasting or exercise, the fatty acids in the pickup from VLDL in muscle are used for energy, right? So fatty acids basically oxidize and utilized for energy. And this is different from the LPL in adipose tissue in fat condition, right? That was used for synthesizing triglyceride, right? Is that clear? Okay. All right, now let's talk about cholesterol. So cholesterol metabolism. First of all, I wanted to review this lipoproteins again. And in the cholesterol metabolism, we basically divided the lipoprotein to the two categories. One called good cholesterol, which is HDL, and the rest of lipoprotein called bad cholesterol, right? We know that LDL and HDL are the major cholesterol-rich lipoprotein, right, and involve the cholesterol metabolism. But recent study also showing that the chylomicron and VLDL also involved in the cholesterol metabolism, although not, like the contribution is not as big as the HDL, LDL, because these two lipoprotein they also affect the cholesterol in the LDL, HDL, right? So in general, HDL is a good cholesterol because it transports cholesterol from peripheral tissues back to the liver for disposal, right? So we can get rid of the excess cholesterol to maintain the balance. And the other bad cholesterol is also called a non-HDL, HDL lipoproteins, which LDL, IDL, VLDL, chylomicron, and altogether called non-HDL. And particularly LDL is a bad cholesterol because it deposits cholesterol into the cells, particularly for macrophages and into the artery wall, right? And contributed to the formation of atherosclerosis. Now let's talk about individual lipoproteins, LDL and HDL. LDL, so core lipids. So the majority of core lipids in LDL are cholesterol esters, right? And also contain the triglyceride. And apoproteins on the surface of LDL is just ApoB100, right? And IDL, which is the VLDL remnant, and core lipids also contain the cholesterol esters and triglycerides. Apoproteins on IDL are ApoB100 and ApoE. So metabolism. LDL, we know the main function of LDL is to transport cholesterol into the cells, right? And this LDL goes into the cells requires LDL receptor. So the uptake of LDL cholesterol is mediated by LDL receptor. So LDL receptor is a key point, right? So here I wanted to make that remind you that ApoB and ApoE, these two apoprotein, ApoB, particularly ApoB100 and ApoE, these two apoproteins are required for LDL receptor binding to the lipoprotein, right? And the second point I want to make that the LDL receptor is expressed mainly in the liver, but also in other peripheral tissue cells. And the number of LDL receptors that determine the rate of internalization of LDL, or in other words, that the LDL cholesterol level in the circulation is primarily determined by the number of LDL receptor, not the LDL level itself. Does that make sense? Because LDL receptor mediates uptake of LDL cholesterol, right? If you have not enough receptor, LDL receptor on the cell surface, then LDL cholesterol cannot be taken up into the liver or other peripheral cells, right? Then you will have more LDL cholesterol remain in the circulation, right? Does that make sense? Yes. Yes, right, right. So for example, because LDL receptor mainly expressed in the liver, right? So liver should be the major, it's important site for pick up the LDL. So if LDL receptor is deficient or whatever reason, then your LDL cholesterol cannot be picked up by the liver, right? So you have a lot of LDL cholesterol remaining in the circulation, right? So again, LDL receptor, the number of LDL receptor is a key that control cholesterol, circulating cholesterol, LDL cholesterol level, right? Okay, so let's finish this LDL receptor slide. So LDL receptor is the single transmembrane glycoproteins. So it means that the located on the surface, cell surface membrane, right, the LDL receptor. And LDL receptor has the site, binding site that can recognize the ApoE or ApoB100 on the surface of LDL glycoproteins, right? So once this LDL receptor recognize the ApoE protein on the LDL glycoprotein, then the whole receptor together with LDL particles being internalized, right, into the cells, and then the whole particle being the glycoprotein being picked up, right? This process is called endocytosis, right? Receptor-mediated endocytosis mechanism, right? Okay, so so this just gives you a little bit details and different version of the LDL receptor, the mechanism, right? So this Y shape is LDL receptor. This is a cell membrane, right? And LDL binds its LDL receptor through the ApoE and ApoB100 protein, right? So then the whole stuff, receptor and LDL particles being internalized, called endocytosis process. And once inside of cells, and then this whole membrane, the receptor actually can be recycled, right, back to the cell surface. And inside of this stuff, like whole particles, that cholesterol can be hydrolyzed, right, to the free cholesterol. And the proteins can be broken down to the amino acids. Right? Questions about the LDL receptors? Okay, so so reacidification of cholesterol and cholesterol ester formation, right? This is the reaction that occurs inside of cell. So once the last slide we already know that the cholesterol ester in the LDL being picked up by the cells, and cholesterol ester being break down to the free fatty acid, free cholesterol, and free cholesterol can be reacidified to cholesterol esters, and this is a reaction that occurs in the cells, right? The cholesterol, one fatty acid molecules attached to the free cholesterol molecules and forming cholesterol esters, and this is enzyme ACAT, right? ACAT is got an acyl-CoA cholesterol acyltransferase, right? ACAT is an enzyme catalyzes the acidification of cholesterol to cholesterol esters. And cholesterol esters is important for the cells. LDL pickup of cells is in normal physiological conditions is also needed, right? Because LDL is bad cholesterol, but doesn't mean that we don't need LDL, right? We need LDL, need cholesterol, but if we have pick up too much, then we're causing the health problem. So this process just in normal condition, right? We need LDL and need cholesterol esters. So cholesterol ester, once being picked up by the cells, can provide substrate for synthesis of bile acids and steroid hormones, right? This is just normal, and also for the for form the core-lipids in the chylomicron and VLDL, and also but here sort of kind of bad things, if too much cholesterol be picked up, then can forming Latin form cells, which is the bad cells that for the atherosclerosis, right? And of course, the cholesterol ester can be hydrolyzed, release free cholesterol and back as needed. So let's see what's the next slide. Okay, I guess we'll just stop here today.